So we did our project on encapsulating enzymes to accurately detect different concentrations of hydrogen peroxide. My name's Philip. This is Joanne, Shlok, Savannah, and Stuti.
So before we get into the details of our experiment, I want to briefly give you guys some context on what a biosensor is, because that is the mechanism we use to conduct our experiment. A biosensor is a device that uses a biological organism, such as bacteria, or a biological compound, such as enzymes or antibodies, to detect the presence and or the concentration of a substance in a solution. Usually the way biosensors work is a colorimetric scale, which means that if you have a lot of a certain substance in a solution, you get a very bright, intense color, like the bright pink you can see to the left. And if you have very little of a substance in a solution, you'll get a very light color, like the one on the right. And how are biosensors able to detect the concentration and portray these colors? It's a three-step process starting with the signal. And the signal is basically the substance that you are trying to measure the concentration of. Once you have the signal, it goes through transduction steps, which is basically what you need to get from the signal to the response. And the response is the final indicator that you use to see how much of the original substance you had. For our experiment, our signal is H2O2, or hydrogen peroxide. And the transduction steps are when H2O2 reacts with Amplex red, another substance, and this reaction is catalyzed by an enzyme called horseradish peroxidase, or HRP. And after the signal and transduction step are complete, we are left with resirufin, which is a substance that colors our solutions pink based on how much of H2O2 was originally present. So what are some applications of biosensors? Currently, one of the biggest applications is for diabetics, who need to monitor their glucose levels constantly. And biosensors provide a relatively inexpensive, non-invasive, and efficient way to monitor glucose levels. On top of that, the food industry is using biosensors a lot to detect pesticides and other contaminants before consumption. Another very interesting application for biosensors is for disease detection and disease screening. This is a picture of cancer because certain tumors are known to release biomolecules that are hallmarks of cancer, certain proteins, certain hormones. And you can use biosensors to detect the presence of these biomolecules in the blood, and then you can see if a patient has cancer and what the development of their tumor is like. And finally, I want to briefly talk about biosensors and their application in the environment. A lot of scientists and a lot of researchers are working on how we can use biosensors to detect certain pollutants and contaminants in the air. So there's a lot of other biosensor applications that I'm not going to talk about, but suffice to say it's a very interesting field with a lot of applications and a lot of promise.
Alright, so first we need to answer the question, why is encapsulation useful? Most of the enzymes that are used in biosensors can unfold and denature in highly acidic or highly basic environments. This renders them useless so they cannot give off a response. So we solve this by using a method called encapsulation. An absorbent gel or bead is placed into an enzyme solution. Once it has absorbed the enzymes are protected inside the nanostructures, and this prevents the denaturation of the enzyme.
This leads us to our research question, which is what method of encapsulation best detects different concentrations of hydrogen peroxide? The significance of our research is based on the applications and impact of hydrogen peroxide. One of many applications of hydrogen peroxide is a pharmaceutical application, such as cleaning scrapes and wounds. Another application is food use, such as washing fresh produce. And dentistry is also a form of application, the clinical application. Hydrogen peroxide may also be used as an antibacterial agent, such as being an additive in milk. Because of hydrogen peroxide's importance in the food and beverage industry, government officials raise the demands for stricter detection protocols. This is why our research is so important. So we hypothesized that in our experiment, we would find that encapsulating the enzyme in agarose gel would provide the best distinction between different concentrations of hydrogen peroxide. We decided to use agarose gel because we learned that through research that in similar studies, agarose gel was commonly used.
So I'm going to go over the experiment that we did to test our hypothesis. So this is just a brief overview, but in step 1, we create the assay solution or the working solution. In step 2, we're going to create the different beads and gels that will be used to soak up the solution that we made in the prior step. In step 3, we're going to make different concentrations of hydrogen peroxide to test the limits of detection. And lastly, we're going to test everything. So in more detail, for step 1, we're going to create the assay solution by putting 15 drops of HRP solution or horseradish peroxidase. We're going to use 10 milliliters of a buffer to keep the enzyme from heating too low of a pH or too high of a pH, which will essentially protect the enzyme. And then we're going to put 3 drops of Amplex Red. In step 2, we're going to prepare the beads and gels that I mentioned earlier. We're going to use polyacrylamide beads, agar gel, iota-carrageenan gel, and agarose gel. And with these, we're going to place the beads and gels in the assay solution for 10 minutes to let them soak up the solution. In step 3, we're going to prepare the different hydrogen peroxide dilutions with the following concentrations. We used 0.0006%, 0.03%, 0.21%, 0.3%, and 0.6%. So as you can see, 0.21% is bolded because this is the amount of hydrogen peroxide that we typically consume in our foods. And from here, we decided to go up and down. We went down to see the limits of detection, and we also wanted to get a gradient of different shades of pink that the Amplex Red will provide. And in step 4, we're going to add 1 milliliter of each hydrogen peroxide diluted solution to the walls in columns 1 through 6, as shown to the picture on the right. And then we'll add the gels and beads to the respective walls, rows A through E.
So the well plate demonstrates our final results. Each row contains different encapsulation methods, and each column contains different concentrations of hydrogen peroxide. The polyacrylamide beads in the first row and iota-carrageenan in the third row only detected the presence of hydrogen peroxide, but did not pick up the differences between concentrations. Agar in the second row was unable to detect the 0.3% and 0.6% hydrogen peroxide concentrations. And agarose in the fourth row not only detected the presence of hydrogen peroxide, but it has darker pink shades as concentration decreased. Lastly, free enzyme solution detected hydrogen peroxide, but the pink shades did not vary in a pattern as the concentrations vary, suggesting that it's not sensitive to the changes in the concentration of hydrogen peroxide. In conclusion, we conclude that the agarose gel was the best way to was most successful at detecting hydrogen peroxide because each concentration is colored and can also accurately picture the differences between concentrations as seen by the clear gradient.
So something really unexpected and really interesting to us that we saw with their conclusions is if you remember back to the first slide when I was explaining how biosensors work, we said that usually if you have more of a substance, you get a brighter, more intense color. And usually if you have less of a substance in a solution, you'll get a lighter, more pale color. And for us, we had the concentration of H2O2 increased as you move from left to right. So you would expect that the shades of pink would get darker as you move from left to right, but instead they got lighter. And the first time we thought it was just a mistake, so we redid our assay, but this is what we got after several trials. And so we're not exactly sure why this happened. Given some more time, we would like to explore this. But one possible idea that we had was that H2O2 is a slightly acidic substance, so maybe it denatured the enzyme, and that's what caused these unexpected results. Talking about unexpected results, I want to briefly go over sources of error. One thing that contributed to some error in our results might be because we were working with some very sensitive materials. I mentioned Amplex Red in the beginning, which is what reacts with H2O2 to start off the signal. And Amplex Red is very sensitive to light. It begins to break down when it's exposed to light. And so it was very difficult for us to manage and try to make sure that Amplex Red was never exposed to light when we were creating our multiple solutions and filling our gels for the assay. On top of that, to some extent, we did have some imprecise measurement. Because we were using gels, we couldn't use a more sophisticated equipment like a spectrophotometer, and we had to eyeball how pink things looked to us. And so this, of course, led to some error in our final results. And lastly, contamination. Like I mentioned, we were dealing with a lot of different solutions, and certain materials are very sensitive, like Amplex Red. And so we tried our best to make sure that nothing was exposed to light, and we were trying to keep everything safe and protected, like you see in that cup, which is completely covered. But that's not always possible, and we acknowledge that that might have factored into our final results. And lastly, for some future directions, in the short term, one of the biggest things we want to do is explore those unexpected results we got, why it got lighter instead of darker as we increased H2O2 concentration. On top of that, we want to test the same solutions in gels with various contaminants. As Savannah mentioned, H2O2 is present in food and in milk, and along with these substances come certain natural contaminants like pesticides. And so this yellow substance over here is a substance we want to use to model contaminants that we might see in food. And we can see how the different gels change their effectiveness based on contaminants. And we also want to test with different ions. So the picture on the right is copper and water, because oftentimes you find copper ions and calcium ions in water. And so testing with these materials and seeing how this changes our gel's effectiveness could also be a next step for us. And in the long term, we want to engineer gels to encapsulate specific enzymes. We were working with horseradish peroxidase, but other people doing other experiments will be working with other enzymes. And so we can try to make gels that are specific and can create the most effective environment for different experiments. So thank you so much for listening to us. Do you guys have any questions?
Well, hydrogen peroxide is a slightly acidic solution by itself, but no, in certain drinks where it might be used for antibacterial uses, they might be very acidic, so biosensors have to be encapsulated.
I think adding on to that, in the lab we were trying to be as sterile as possible, but when someone might be using this in their own home, the conditions might not be that perfect or ideal, and so testing with encapsulation just makes it more effective and more accurate overall.
Hydrogen peroxide is an additive in milk, but I believe it would be used more like the food and beverage industry than like the actual people.
Does that answer your question? I think if we go back to that, if you guys remember that one slide that Philip talked about, yeah, right here. So the average amount of H2O2 we're exposed to is 0.21%, which is already not a lot. And our sensor can detect down to 0.0006%, which is significantly less. So our sensor is really sensitive and it can pick up on even the smallest concentrations of H2O2.
Well, by definition, the polyacrylamide encapsulated the enzyme the best, but since it encapsulated so much enzyme, it wasn't very sensitive to the different changes in concentration. So I think agarose gel was the average, the fine line between too much and too little. So in our future directions, we wanted to couple gel with an instrument to give quantitative results. Unfortunately, we don't have that kind of technology right now, and with the time frame we were given, we didn't really have enough time to do that. We were thinking of maybe using a modified version of a spectrophotometer. Well, the level that we had in our slide, which is 0.21%, it's not very toxic to normal to adults, but to children and people with certain conditions, it can be more toxic.
Thank you.